morning, everyone. Today we're going to talk about fatty acid oxidation and ketogenesis, as well as cholesterol and bile acids. So the first portion will be about fatty acid oxidation and ketogenesis, and our objectives are to explain fatty acid oxidation and the role of carnitine in fatty acid transport. We also want to understand the clinical significance of fatty acid deficiency and understand ketogenesis and its clinical significance. Fatty acids, in order to be oxidized in the mitochondria, must first be activated in the cytoplasm. And this occurs through the enzyme fatty acyl-CoA ligase. And this has a number of names. In this graphic, it's shown as acyl-CoA synthetase. And it also can be called thiokinase. Okay? So this reaction, where we're activating the fatty acid in the cytoplasm, requires 2 ATPs. You can see 1 ATP is used here in the acyl-CoA synthetase reaction. And then we also have 1 ATP that's used to hydrolyze this, this diphosphate in order to ensure irreversibility of the reaction. So we can see that this is just a one-way reaction here. And this is the combination of our fatty acid plus our CoA to produce fatty acyl-CoA. Okay, so in this next graphic, it shows how fatty acids are transported into the inner mitochondrial membrane in order to be oxidized for energy. So for this top part, it's just what we just showed, fatty acid. We have our ATP and our CoA. They go through this acyl-CoA synthetase enzyme in order to produce fatty acyl-CoA. And this can go through the outer mitochondrial membrane, but not the inner mitochondrial membrane. So the transport into the inner mitochondrial membrane is accomplished via an acyl-CoA carnitine intermediate. So here we have, So here we have our carnitine here, which combines with our acyl-CoA here, and it generates acyl-carnitine using the enzyme CPT1. Then the acyl-carnitine molecule is able to go through the inner mitochondrial membrane of the mitochondria through the action of carnitine acyl-carnitine translocase, the CAT here. So we get in, now we're able to get into the inner mitochondrial membrane where we need to be. So then this molecule, acyl-carnitine, goes through another enzyme, CPT2, which separates the acyl-carnitine back into our CoA, our carnitine, and our acyl-CoA. And this fatty acyl-CoA can then go through beta oxidation in order to produce energy, which we're going to go into a lot more detail about. And the carnitine can go back out of the inner mitochondrial membrane in order to shuttle another fatty acyl-CoA across. Okay, so beta oxidation, I'm sure, is a term that you're familiar with. And this is the process by which there is a removal of 2 carbon units from the carboxyl end of a fatty acid in order to oxidate the fatty acid. So in beta oxidation, we remove 2 carbon units from the end, and these 2 carbon units are in the form of acetyl-CoA. Okay, and then this should kind of ring a bell. Acetyl-CoA is our common substrate for entering the TCA cycle. So once we break off this 2 carbon unit, it can then go into the TCA cycle to produce energy. And the remaining fatty acid continues to undergo beta oxidation, where these 2 carbon units are lysed off until the fatty acid is completely oxidized. Okay, so that's shown here. This is kind of a busy slide, but we're going to work our way through it. Okay, so the first step, so in this example, it's palmitoyl-CoA, which is a 16-carbon fatty acid. But this process applies to beta oxidation for all fatty acids. So we have our palmitoyl-CoA, and then we have an oxidation step through the dehydrogenase enzyme to produce FADH2. Okay, so we're taking some of the hydrogens off of here, placing them on the FADH2. Then we have hydration. Then we have another to produce beta-hydroxyacyl-CoA here. Then we have another oxidation step with a dehydrogenase enzyme to produce an NADH. And then in our last step, we have the cleavage of this beta-ketoacetyl-CoA, okay? So we have this cleavage via a thiolase enzyme. And our products are this 2-carbon unit of acetyl-CoA, which can go to the TCA cycle. And then we have the rest of this fatty acid, which is 2 carbons shorter, so it's just 14 carbons. And then it goes through this whole process again, so that it'll have another acetyl-CoA, and we'll have a 12 carbon, which will go back through, and so on and so forth. So for each round of beta oxidation, where we're removing 2 carbons, we produce one FADH2, one NADH, and one acetyl-CoA. However, if you think about it, when we get down to our last 4 carbons, we really just need a 1 round of beta oxidation to separate those 2 into 2 acetyl-CoAs. So the amount of acetyl-CoAs we get from oxidation of our fatty acid is really the number of rounds of beta oxidation plus 1. And hopefully this will become more clear when we move forward. Okay, so for beta oxidation, we get each round of beta oxidation, 1 NADH, 1 FADH2, 1 acetyl-CoA. And then we'll get an additional acetyl-CoA for that last one that's just kind of left. So, and then the acetyl-CoA goes to the TCA cycle, and this is just a reminder from earlier in the course, where it's generated, where it's oxidized, and we get 3 NADH, 1 FADH2, and 1 ATP. So all of this together is, we'll remember that NADH is 3 ATP, so this is 9 ATP, plus 2 is 11, plus 1 is 12. So each round of the TCA cycle produces 12 ATP. Okay, so let's look at how many ATP are produced from a fatty acid. So let's use a fatty acid with 18 carbons. Okay, so for each slash is a round of beta oxidation where we're lysing off 2 carbon units, okay? So 1, 2, 3, 4, 5, 6, 7, 8, okay? So we have 8 rounds of beta oxidation. And we get 5 ATP per turn, because remember we get 1 NADH and 1 FADH2. So for 8 turns of beta oxidation times 5 ATP per turn, we get 40 ATP just through the beta oxidation part. And then our acetyl-CoA, CoAs that are produced from beta oxidation go into the TCA cycle. So let's count our 2 carbon pairs, and we get 9. 1, 2, 3, 4, 5, 6, 7, 8, 9, or 18 divided by 2. So 9 moles of acetyl-CoA times 12 ATPs per turn of the TCA cycle, we get 108 ATPs. And remember from our first step, we use 2 ATPs just to activate the fatty acid. So in total, we get 146 ATP from this 18 carbon fatty acid. Okay, now we know that fatty acids are more energetically dense compared to carbohydrates. And this means they yield more energy per carbon atom compared to carbohydrate oxidation because they're further reduced. They have more hydrogen atoms that can go through the TCA cycle, and also they go through beta oxidation to produce this additional energy. So in our 18 carbon fatty acid, we saw that it produced 146 moles of ATP. However, 18 moles of carbon in the form of glucose only produces 114 moles of ATP. So we can see that the fatty acid is more energetically dense. It produces 8 ATP per carbon compared to 6.3 ATP per carbon in a glucose molecule. Now, in nature, most of the lipids we encounter are in the form of, have an even number of carbon atoms, so C18, C16, C20. However, we also have some fatty acids that have an odd number of carbon atoms, and this makes for a little bit different energy production. So upon beta oxidation, when these odd number of carbon fatty acids go through, we still are going to lyse these 2 carbons off okay, to produce acetyl-CoA. But then at the end, we have a 3-carbon unit left, which is this propenyl-CoA. And we've talked about that in the class earlier on, okay? So this propenyl-CoA can then be converted to succinyl-CoA in an ATP-dependent pathway. So it uses one ATP to get to the succinyl-CoA. But then the succinyl-CoA can go into the TCA cycle for oxidation. Okay, so let's look at this example where we have 21 carbons. So again, we're lysing it every 2, okay? So we have 9 turns of the beta oxidation times 5 ATP per term to get 45 ATP from the beta oxidation of this molecule. And then we count our 2 carbon units of acetyl-CoA. So this last one we're not counting because this is the propenyl-CoA. And so we get 9 moles of acetyl-CoA for 108 ATP once it goes through the TCA cycle. And then we're left with our propenyl-CoA. And to be converted to succinyl-CoA, it uses 1 ATP. But then succinyl-CoA goes through the TCA cycle and produces 6 ATP. And of course, in the beginning of our activation of fatty acids, costs 2 ATP. So we would get 156 ATP from the total oxidation of this carbon-21 fatty acid. Okay, so many of the problems that we see in fatty acid metabolism are associated with deficiencies in some of these substrates or enzymes that are needed for the oxidation process. So the most common is a deficiency in carnitine. We may also see deficiencies in the CPT1 or CPT2 enzymes, as well as in the acetyl-CoA dehydrogenase enzymes. Now, when there's not enough carnitine, we aren't able to shuttle our fatty acids into the inner mitochondrial membrane where they need to be for oxidation. Okay, and this can occur in newborns, and I meant to put in particularly in premature infants. Okay, and also it can occur in patients undergoing hemodialysis or exhibiting organic aciduria. Symptoms can be highly variable from muscle cramping to severe muscle weakness or even death, but this can be treated by oral carnitine. Okay, so next we're going to talk about ketogenesis. Okay, so when we have a large amount of beta oxidation, we produce a lot of acetyl-CoA. But remember, we don't only need acetyl-CoA to go through the TCA cycle. We also need the other substrates. So at the end of the TCA cycle, we have oxaloacetate that needs to combine with the acetyl-CoA to keep the system kind of going. But if we don't have those substrates, then the acetyl-CoA starts to build up and to be converted into ketone bodies. And these ketone bodies are acetoacetate, beta-hydroxybutyrate, and acetone. Okay, so here we're going to talk about how these ketone bodies are formed. Now, in our last reaction, remember we had our fatty acid and, we had the reversal of this first reaction. So we went from acetyl-CoA to acetyl-CoA and the rest of our fatty acetyl-CoA. So that was a thiolase reaction. But you can see here the thiolase reaction goes both ways. So when we're producing our ketone bodies, we actually have the condensation of 2 acetyl-CoAs via this thiolase enzyme to produce acetyl-CoA. Then we have the addition of another acetyl-CoA through the action of HMG-CoA synthetase in order to produce HMG-CoA. Okay, so and this enzyme, the HMG-CoA synthetase, is found in large quantities in the liver. Now, some of this HMG-CoA is going to be shuttled out of the inner mitochondrial membrane for cholesterol synthesis. But in this process that we're talking about here, our HMG-CoA is going to stay in the mitochondria, and it's going to be converted into acetoacetate through the action of a HMG-CoA lyase. Sorry, I'm realizing I'm doing 2 screens now, which I don't usually do, so I'm realizing I might not be pointing as I'm going. Okay, So we have our production of acetoacetate here, and this can undergo spontaneous decarboxylation to acetone, or it can be enzymatically converted to beta-hydroxybutyrate through the action of this dehydrogenase enzyme here. So the acetone, the acetoacetate, and the beta-hydroxybutyrate are all of our ketone bodies, okay? And which are produced when we build up too much acetyl-CoA from beta oxidation. Okay, so as I mentioned earlier, when we don't have enough carbohydrates, we don't have enough oxaloacetate, we can't keep running the TCA cycle, we get this buildup of ketone bodies in the liver. And the good part about the ketone bodies is that they can be used as fuel by other tissues even when we don't have enough carbohydrates. So in the early stages of starvation, when we oxidize our last fats, our heart and skeletal muscles can rely on ketone bodies for energy in order to preserve our precious glucose for our brain, which is really has a real specific need for glucose. So the ketone bodies are utilized by the extrahepatic tissues by converting the ketone bodies back to acetyl-CoA. So it's kind of confusing because we're condensing the acetyl-CoA to get to our ketone bodies, and then it kind of is shuttled out to these extrahepatic tissues where it's converted back to acetyl-CoA. Okay, so this is what this is showing here. So the first step in metabolizing the ketone bodies that can be used by the extrahepatic tissues for energy is conversion of this beta-hydroxybutyrate to acetoacetate and of acetoacetate to acetyl-CoA, okay? And so the first step involves the reversal of this beta-hydroxybutyrate dehydrogenase reaction. So it can go in both ways. It's a reversal of what we just saw. And then the second step is involves the action of this ketoacetyl-CoA transferase. You may also see it as the acetoacetate to succinyl-CoA transferase. Okay. And then the last step is this thiolase reaction again, where we're converting the acetyl-CoA back to two acetyl-CoAs. And then this can be utilized by the extrahepatic tissues. Okay. So we have the buildup of our acetyl-CoAs, conversion to ketone bodies, shuttle to other parts of the body, conversion back into acetyl-CoA. Okay, so in normal fed person with normal metabolism, we have very low production of ketone bodies. But when we have shortages in carbohydrates, this can cause the liver to produce ketone bodies from acetyl-CoA during fatty acid oxidation. And we see this in diabetes, we see this in general starvation, or when people have a very low carb diet. And it's kind of a two-sided coin, I guess I should say, because it allows some of our tissues to use ketone bodies for energy and preserve the limited glucose that we have for the brain. However, it can also, because these ketone bodies are acidic, it can also disturb the body's acid-base balance, and this has consequences. Okay, so the most common time we see the ketoacidosis is in untreated insulin-dependent diabetes. And this is termed diabetic ketoacidosis. So this results when the when we have a reduced glucose supply because the glucose cannot get into our cells because of low circulating insulin, and then because our body is not getting the glucose, it's saying, okay, it's producing glucagon and saying, let's break some of this down, I need some energy. So we have an increase in fatty acid oxidation. So decreased glucose, increased fatty acid oxidation. So when we have the increased fatty acid oxidation, we get increased acetyl-CoA production. And increased ketone body production that exceeds the ability of the peripheral tissues to oxidize these ketone bodies. So we start to get the buildup of these ketone bodies. Now, because these are acids, they lead to a decreased pH and blood acidification, which is primarily dangerous because it impairs the ability of hemoglobin to bind oxygen. So it compromises our oxygen delivery. And this is why we see the rapid shallow breathing in diabetic ketoacidosis. The keto acids can also induce nausea and vomiting, and this can lead to fluid and electrolyte loss. So if we'll kind of remember back, there's for other reasons of osmolarity, of high glucose concentration in the blood, this also leads to a lot of fluid loss. So we're kind of having a compounding issue here where we're losing a lot of fluid in someone, or someone who has DKA is losing a lot of fluid. Okay, so this hyperosmolarity of the glucose plus the dehydration that we're getting from all this fluid loss plus the acidification of the blood leads to increased osmolarity in brain cells, and this can alter consciousness and is responsible for the cognitive changes that we see in diabetic ketoacidosis. Okay, and then this is just kind of an overview of what we just talked about for ketone body production and utilization. So we have our fatty acid, we have conversion to acetyl-CoA, beta oxidation, eventual conversion to our ketone bodies. The ketone bodies are shuttled to our extrahepatic tissue, where then they can be reconverted back into acetyl-CoA and go through the TCA cycle here. But if we don't have the ability to go through the TCA cycle, these can build up. And here are our practice questions one, so I'll let you do those on your own. And we're going to move on to cholesterol synthesis and bile acids. All right, so the objectives of this portion of the lecture are to explain cholesterol biosynthesis, as well as the regulation and utilization of cholesterol. We also want to understand bile acid synthesis and utilization, and be familiar with the clinical significance of bile acid synthesis. So we've talked some about cholesterol, and this is an extremely important biological molecule in that it's imperative for membrane structure, and we can see that in this little graphic here, that we have our cholesterol molecules that provide structure in our cell membranes in the phospholipid bilayer. And also cholesterol is a precursor for steroid hormones and bile acids, both of which have their own crucial functions that we'll talk about. So dietary cholesterol, so cholesterol that we get from the diet and that we synthesize endogenously, are transported through circulation in lipoprotein molecules. And we talked about that last week, our VLDL, our LDL, and so on. And also, okay. And cholesterol is stored in the cells as cholesterol esters. Okay, so the synthesis and utilization of cholesterol needs to be tightly regulated. So we definitely need to have enough of it in order to maintain our cell membranes and produce these like steroid hormones and bile acids. But if we have too much of it, we can have an overaccumulation and deposition within the body. And we're all familiar with this situation here. So we have our healthy artery here, and then we have our overaccumulation of cholesterol and cholesterol-rich lipoproteins in the endothelial lining of the coronary arteries. And this leads to atherosclerosis in coronary artery disease. Okay, so a little less than half of the body's cholesterol is synthesized endogenously, and the remainder we get through the diet. Most of our cholesterol is synthesized in the diet or in the liver, though it's also produced in the intestines, and it can be produced in small quantities in nearly all of our tissues. Synthesis of cholesterol occurs in the cytoplasm with enzymes from the cytoplasm, microsomes, and perioxomes. Now, the first step of cholesterol synthesis is that we need to have a precursor of acetyl-CoA, sounds familiar, which is derived from the oxidation reaction in the mitochondria, either from our fatty acids or from pyruvate. And the acetyl-CoA is then transferred into the cytoplasm. Okay. So acetyl-CoA must be available in the cytoplasm in order to be utilized for cholesterol synthesis. And it enters the cytoplasm in the form of citrate, okay? So we have citrate here. Then it's converted to oxaloacetate, and then to eventually to acetyl-CoA through, and then eventually to acetyl-CoA. Okay, I'm sorry, let me back up a little bit. So we get our acetyl-CoA from our oxidation reactions in the inner mitochondrial membrane. It is converted into citrate, and then it can be shuttled out into the cytosol, okay? And then it's converted to oxaloacetate and acetyl-CoA, and it uses 1 ATP in this reaction. Okay, so cholesterol biosynthesis has a lot of different steps, and we're just going to talk about kind of the main reaction stages, of which there are 4. So the first one is our really important regulatory stage, and this is the synthesis of mevalonate from 2 acetyl-CoAs. Okay, so acetyl-CoA is converted to mevalonate through a series of reactions. And it begins with this thiolase reaction, and then the production of HMG-CoA here through HMG-CoA synthetase. So this thiolase reaction, again, we've seen before, where we form acetyl-CoA, and then a third mole of acetyl-CoA is introduced here in order to produce this HMG-CoA. Then the HMG-CoA undergoes a reaction with the enzyme HMG-CoA reductase, and this is kind of what we think of as the rate-limiting step in cholesterol biosynthesis. So this is the same number of these first steps are the same as in ketone body production. So if you'll remember back when we were going through our ketone body formation slide, I said, okay, and then the HMG-CoA can also get shuttled out and be used for cholesterol synthesis. So these first steps are the same, but then the HMG-CoA is catalyzed by HMG-CoA reductase to produce mevalonate. So that's step 1. And this HMG-CoA reductase is subject to strict regulatory controls that we're going to talk about. So the next steps, I'm just going to summarize quickly, where mevalonate is converted to 2 activated isoprenes. I did it again. Sorry, I've been moving my mouse on my own screen instead of the second screen. Okay. It's converted to 2 activated isoprenes. Then these are condensed to form squalene, and then squalene is the precursor to cholesterol. So, but the main stage that we need to remember is this first stage, and the main step in this first stage that we need to talk about is this HMG-CoA reductase, because this is the major rate-limiting step, and the one that's highly regulated. Okay, so in normal healthy adults, we produce about 1 gram per day of cholesterol and use about 0.3 grams per day. And we maintain a relatively constant blood level by controlling our endogenous synthesis of cholesterol. So our regular constant blood level in a healthy range is considered to be 150 to 200 milligrams per deciliter. And however, our synthesis, the regulation of our synthesis, is somewhat regulated by how much cholesterol we take in. So both dietary and synthesized cholesterols are used for the purposes that we discussed earlier. But the greatest proportion of our cholesterol is used for bile acid synthesis. Okay, so our cellular cholesterol supply is regulated by three distinct mechanisms. So the first is the one we're going to focus on the most, the regulation of HMG-CoA reductase activity and levels. And this is the primary means for controlling the level of cholesterol biosynthesis. Second, we can regulate our cholesterol synthesis by regulation of intracellular free cholesterol through the ACAT enzyme that we talked about last week. Also, we can regulate plasma cholesterol levels via our LDL receptor mediated uptake. That says medicated. And I'll put the, I'll post the correct one for you. Okay, mediated reverse transport. So how, what is our ability to pick up the cholesterol and get rid of it? Okay, so not only do we have three distinct mechanisms to regulate cholesterol biosynthesis, we also have three distinct mechanisms to regulate HMG-CoA reductase regulation. And remember, this is our most important point of cholesterol synthesis regulation. So the mechanisms are the control of gene expression of this enzyme, the rate of degradation for this enzyme, and most importantly, our phosphorylation or dephosphorylation of this enzyme to make it either active or inactive. So the HMG-CoA reductase is the most active when it's dephosphorylating, doesn't have the P on it. So when we add the phosphate group onto it, it decreases activity. And this phosphorylation is regulated by AMP-regulated protein kinase, or AMPRK, which is itself activated by phosphorylation of an enzyme called kinase. So this all gets a little confusing, but we'll go through a graphic of this. And so this phosphorylation, dephosphorylation is regulation of the HMG-CoA reductase through regulation of a covalent modification via this phosphate group. Okay, so let's just kind of review what we just talked about. So we have our HMG-CoA reductase in its active form. When we add the phosphate group, it's in its inactive form. So phosphorylation decreases activity. This phosphorylation is regulated by AMP-regulated kinase, okay, here. And the AMP-regulated kinase is active in its phosphorylated form, inactive in its dephosphorylated form, and this activity is regulated by the enzyme kinase. So adding a kinase means adding a phosphate on, so it's adding a phosphate on to the group that will add a phosphate on to the HMG-CoA reductase. That's why it's called kinase. So I'm going to read through the next slides and kind of go through them so you'll have them in narrative form, but I'm going to stay on this slide just so that I can kind of point and move through. So, so we have our HMG-CoA reductase here, inactive form regulated by AMP-RK, which is regulated through kinase here. Okay. So this activity of HMG-CoA reductase, so we've kind of gone through all of these up here, is further regulated through the secondary messaging system of CAMP. Here. We've always got CAMP, it seems like. Okay, so increased CAMP that we get from stimulation from glucagon or epinephrine is going to lead to the activation of CAMP-dependent protein kinase A here. Okay, so pKa phosphorylates and activates. So it's a kinase, so it adds a phosphate group on, and it's going to phosphorylate and activate phosphoprotein phosphatase inhibitor 1, so PPI1 here. Okay. Now, this PPI1 can inhibit activity of numerous phosphatases. So this includes, so we have so think of this as a phosphatase inhibitor. It's inhibiting taking off a phosphate, okay? So this has action in this regulatory mechanism in two different places. So first, this PPI1 can inhibit this protein phosphatase 2C here, okay? So if it's inhibiting this phosphatase here, then we're going to stay, the AMPRK is going to stay phosphorylated and it's going to stay active, okay? So if we think about this in the way of hormone regulation, which again we'll touch more on, but so glucagon and epinephrine stimulate CAMP, right? Okay, so that means, so what those hormones are saying is, I don't have enough, I need to use whatever I have, we don't want to build up anything, okay? So in that way, we're saying, okay, let's inhibit this so that we keep this active, this AMPRK active, so that we keep this HMG-CoA reductase inactive. So we're not going to build any more cholesterol. Makes sense. We're in stress, we're not going to build cholesterol. Now, this PPI1 also inhibits the HMG-CoA reductase phosphatase. So this phosphatase is responsible for taking the inactive form and moving it to the active form. Okay, so this enzyme would actually make it so that the HMG-CoA reductase was active. All right, and we would build it. But if we're inhibiting this, then we're going to stay in this inactive form. So again, we're under stress, glucagon and epi. We don't want to build anything, so we're going to keep HMG-CoA reductase in its inactive form. Oh, we just went through all that. Okay. And we talked about hormonal control via CAMP. So insulin decreases CAMP and activates cholesterol synthesis. So it's kind of an anabolic enzyme. We're building cholesterol. While glucagon and epinephrine increase CAMP and inhibit cholesterol synthesis. Now, cholesterol synthesis is also regulated by cholesterol supply. So if we have a lot of cholesterol from excessive dietary consumption or from a genetic issue that makes it so we produce a lot of cholesterol, then that's going to increase the rate of the degradation of the HMG-CoA reductase that we need for cholesterol biosynthesis. So it's kind of a feedback mechanism. Okay. So the cholesterol is transported in the plasma primarily as cholesterol esters in lipoproteins. So the cholesterol is synthesized in the liver or comes from dietary sources. And when the cholesterol that we get from these two places exceeds the needs, it's transported into the serum by VLDLs, which are then converted to other lipoproteins via lipoprotein lipase. The cholesterol that's found in plasma membranes can then, this is kind of a review from last week, can then be extracted by HDLs and esterified by the HDL-associated enzyme LCAT. And ultimately, the cholesterol is then shuttled back to the liver, where it's converted to bile acids and can be excreted in the bile as free cholesterol or bile salts. So the end products of cholesterol utilization are bile acids, and these are synthesized in the liver, and are really our major method of being able to excrete cholesterol or get rid of cholesterol through our feces. So, however, cholesterol excretion in the form of bile acids is insufficient if a person is consuming way too much cholesterol. So it really only works to excrete cholesterol up to a certain rate, and then we're going to have the buildup of cholesterol in our arteries. So the most abundant bile acids are chenodeoxycholic acid and cholic acid. So before bile acids leave the liver, they're converted to bile salts. And this happens through the conjugation of the bile acid with either the glycine or taurine amino acids before their secretion into the bile caniculi. So we just mentioned our two major forms of bile acids, okay, the cholic acid and the chenodeoxycholic acid. And then these combine with either glycine or taurine, and then we get 4 major bile salts that are listed here. And the glycine forms of the bile salts outnumber the taurine forms of the bile salts by approximately 3 to 1. Okay. Now, since the carboxyl group is more water soluble when the hydrogen ion or the amino acid is separated from it, bile salts, which have the amino acid completely separate from the carboxyl group, are more water soluble compared to their precursor bile acids. And that's because the bile acids have the hydrogen ion that likes to stick to the carboxyl group, so it makes it less water soluble. So because bile salts are more water soluble, they are a more effective emulsifier of fats in a water solution. And therefore, they're more effective at mixing fats with the water-soluble enzymes in digestion. And we know this because we see the bile salts that are primarily used for micelle formation. So after the bile salts are produced in the liver, they either flow from the bile, produced in the liver, they flow from the bile duct into the first part of our small intestine, the duodenum, for immediate use in digestion for micelle formation, or they're stored in the gallbladder to be saved for future digestive needs. So 95% of the bile acids are reabsorbed in our last part of our small intestine, which is our ileum. Okay, and where they travel through the blood, attached to the blood protein albumin. Okay, so it's absorbed in the ileum, absorbed back into our bloodstream, attached to albumin, and then they go back into the liver. So this is kind of our enterohepatic circulation of bile salts. So about 15 to 30 grams of bile salts are circulated in this sequence each day. About 0.5 grams are lost in the feces, and about 0.5 grams are synthesized endogenously in the liver. So you can see that each day the amount of bile acids is equivalent to that, or produced is equivalent to that lost in the feces. So that the pool of bile acids is constantly maintained. We have this kind of homeostasis that's accomplished by a system of feedback control. Okay, so bile acids have 4 physiologically significant functions. So their synthesis and excretion in the feces represents really the only significant mechanism for eliminating excess cholesterol. Oh, I'll say number 1, but I think you can get this 2, 3, 4. Also, bile acids and phospholipids solubilize cholesterol in bile, preventing cholesterol from precipitating in the gallbladder and building up and causing problems. Bile acids also emulsify fats in order to facilitate digestion of the fats by increasing accessibility of digestive enzymes such as pancreatic lipase. And because they help digest fats, they also facilitate the digestion of fat-soluble vitamins. Okay, now we're going to kind of shift gears to another product of cholesterol. So we saw that they produced bile acids and salts, but they also are used as precursors for steroid hormones. And all steroid hormones, almost all steroid hormones, are derived from cholesterol, except for retinoic acid, which is from vitamin A. They also all contain a same basic structure as cholesterol, except for vitamin D. All steroid hormones, I should say, except for vitamin D. So the conversion of the 27-carbon cholesterol to the 18, 19, or 21-carbon steroid hormones involves a rate-limiting, irreversible cleavage of a 6-carbon residue from cholesterol to produce a substrate called pregnenolone, which is a 21-carbon. So again, we have our 26, we cleave off a 6-carbon, and we produce this 21-carbon substrate. Okay, and then in this graphic, we just see how our exactly the pathway for producing these steroid hormones. So here we have cholesterol. We just talked about that it's converted to pregnenolone, and then pregnenolone ultimately is converted to progesterone, aldosterone, cortisol, estrone, testosterone, and estradiol. Okay, so these are all very important hormones, including our sex hormones, that have very crucial functions in our body, and they are all synthesized from cholesterol. Okay, and then this slide just shows, demonstrates kind of the basic description of some of our very important steroid hormones. Okay, so pregnenolone is kind of our intermediate for producing our important steroid hormones. Progesterone is responsible for changes associated with the luteal phase of the menstrual cycle and a differentiation factor in mammary glands. So when we say differentiation factor, breast tissue can have be very characteristically different, and now we're kind of looking in, or I should say researchers are looking into how this differentiation could impact risk for breast cancer. We have aldosterone, which is responsible for raising blood pressure and fluid volume and increasing sodium uptake. We have testosterone, which is responsible for the secondary male characteristics. We have estradiol, which is responsible for secondary female sex characteristics. And we have cortisol, which is kind of one of our major stress hormones. And it's associated with elevated blood pressure and sodium uptake, and has an impact on the immune system. Okay, and then for our last topic, we're going to talk about plant sterols and stanols. And we include this in this section, not because these are produced at all from cholesterol, but because they have a structure that's very similar to cholesterol. So these are produced only by plants, not by animals or humans. We are animals. So these sterols and stanols are found in various plant matter, including wood pulp, leaves, nuts, vegetables, vegetable oils, corn, rice, and other plants. The major plant sterol that is found in the diet is from soybean, and it's called sitosterol. And as we would assume, people who consume more of an Asian-based diet or a strict vegetarian diet have much higher intake of this plant sterol. Okay, and as I said, they have a similar structure to cholesterol, but they have a methyl or ethyl group on their side chains, and this decreases their absorbability. So they're barely absorbed or not absorbed at all compared to cholesterol, which we do absorb. So what happens, actually I'm going to leave this to the next slide because we have a little YouTube video that shows how plant sterols and stanols can work to decrease cholesterol absorption. But basically, they compete with cholesterol for absorption because they look so much like cholesterol, and therefore we have lower rates of cholesterol absorption, which ultimately lowers LDL cholesterol. So less cholesterol is absorbed, less cholesterol returned to the liver, increased hepatic LDL uptake because we have less of it, and therefore we have decreased serum LDL cholesterol levels. So let's watch this little YouTube video that demonstrates how these sterols and stanols work. Do you guys see this?
High LDL cholesterol is widely accepted to be a major risk factor for coronary heart disease, which is a leading cause of morbidity and mortality around the globe. As part of normal metabolism, the liver releases cholesterol into the bloodstream. A member of the lipids family and an essential molecular building block in the human body, cholesterol circulates around the bloodstream, attached to proteins, forming complexes known as low-density lipoprotein cholesterol, or LDL. In this form, cholesterol is carried to the tissues that need it. However, too much LDL can be problematic, as it can lead to the formation and buildup of atherosclerotic plaques on artery walls. This can cause problems such as obstructions and ultimately myocardial infarction. Cholesterol present in the gut is contained within bile from the liver and from food sources in the diet. In order to be absorbed from the gut into the bloodstream, cholesterol mixes with bile salts, fatty acids, lecithin, and triglycerides to form tiny water-soluble packets called dietary mixed micelles. The micelles then make contact with the intestinal cell wall, where their contents are taken up into the cell. Once inside the cell, the micelle contents are repackaged into spherical particles called chylomicrons. The chylomicrons then carry their fatty contents into the bloodstream, where they are delivered to their target tissues, including the liver. Absorbed and newly synthesized cholesterol is then released from the liver cells into the bloodstream, where it gradually forms LDL cholesterol via the transformation of other lipoproteins. However, there is an ingredient called plant sterols, which interferes with the absorption of cholesterol from the gut and significantly reduces the amount of cholesterol that reaches the bloodstream. Strong evidence suggests that plant sterols displace cholesterol from the micelles formed in the gut, which are essential for cholesterol absorption. As a result, less cholesterol is contained within the micelles, and cholesterol absorption into the bloodstream is reduced by as much as 30 to 40%. The cholesterol that is not incorporated into micelles is subsequently excreted from the body along with plant sterols, which are pumped back into the gut lumen. A wealth of scientific evidence clearly indicates that a daily intake of 2 to 2.5 grams of plant sterols results in a reduction of intestinal cholesterol absorption of 30 to 40%, which leads, on average, to a reduction of 10% in LDL cholesterol levels. This 10% reduction is important because evidence suggests it can cut the risk of coronary heart disease by as much as 20%.
Okay, so that is actually all. Hopefully you saw that. Sorry, I'm getting adjusted to this computer, but otherwise you have the link in your PowerPoint slide. But that is all that I have for you today about fatty acid oxidation and ketosis, ketogenesis, and cholesterol and bile acids. Have a good week, everyone.